Hello students, in this video I will explain you October-November 2017 series paper 62 alternative to practicals. Let us discuss question number 1. Question number 1. Starch is an important food source that is digested by the enzyme amylase to form the reducing sugar maltose. You can see this chemical equation given here. Right, starch word equations. Yeah, starch plus water, amylase catalyze it to form maltose. Maltose is a disaccharide. Okay. Some students investigated the effect of enzyme concentration on the rate of digestion of starch. Effect of enzyme concentration on the rate of digestion of starch. Step 1. 3 test tubes were labeled A, B, C. Step 2. 5 cubic centimeter. This is the volume of starch solutions was put into each of the test tubes A, B and C. Step 3. Another 3 test tubes were labeled as A1, B1 and C1. Step 4. 1 cubic centimeter of 3% amylase solutions was put into test tube A1 and 1 cubic centimeter of 2% amylase solution, and then in B1 and 1 cc of 1% amylase solutions was put into test tube C1. So you can see the percentage concentration of amylase solutions, right, changes 3, 2 and 1. So that is the purpose, to investigate effect of enzyme concentration on rate of digestion of starch. Now, step 5, all 6 test tubes were placed into a water bath at 60 degrees Celsius for 3 minutes. Okay, let's move to step 6. A white tile was divided into 3 sections. They are labeled as A, B and C as shown in figure 1.1. Step 7. Iodine solution. Now, iodine solution was dropped onto the tile to form 2 rows. You can see here, this to form rows of 10 drops, approximately the same distance apart. Rows of 10 drops, you can see there are 2 row 1 and row 2, right? So in each of the sections A, B and C, there are 2 rows in each of the sections, and there are 10 drops in each row. Step 8. A glass rod was dipped into starch solution in test tube A. To remove some of the solutions, the glass rod was then touched onto the surface of first drop and then the second drop. Of iodine solutions in row 1. on the sections of the tile labeled A. The glass rod was rinsed and dried. Now, step 9. Step 8 was repeated using the amylase solutions. First they use starch solutions and then amylase solutions in test tube E1 and drops of iodine solutions in route 2 on the sections of the tile labeled A. Now here you can see in step 8, okay, same step was repeated, right, by using amylase solution now in test tube A1 and drops of iodine solutions in route 2. Okay. Step 10, a timer was started and the amylase solution in test tube A1 was poured into test tube 1, test tube A. So as you know that test tube A1, it contains amylase enzyme and test tube A that contains starch solutions. Now they are mixed. So mixture of starch and amylase in test tube A was stirred with a glass rod and then some of the mixtures was immediately removed using the glass rod. The glass rod was then touched onto the surface of first drop and then second drop of the iodine solutions in the row 3 on the sections of the tile labeled A. The glass rod was rinsed and dried. So now you can see in row 3, this is row 1, this is row 1, this is row 2, this is row 3, right? So row 1 and row 2, there in row 1 only starch solutions was taken from test tube A and test, and in row 2 only amylase enzyme solutions was taken by glass rod and test on this, and in row 3 you can see amylase and starch both. Now in first row only starch was there, right? So it will turn to blue black, and row second only enzyme is there but there is no starch, so it will remain brown or yellow or light brown, right? Row 3, right, here the color will change. Here enzyme and starch both are taken, so enzyme will digest the starch, right? So the color will remain yellow brown or light brown. Now after 1 minute, the glass rod was used to remove some of the mixture from test tube A, okay, and touched onto the first drop and then second drop of row 4. Now here after 1 minute, from here, I'll take the red color here. After 1 minute, this fourth row, after 1 minute, it was touched. Okay, so within 1 minute, how much starch is digested? Okay, then the color will show. Here the starch is there because immediately the starch starch and amylase solution was taken, so digestion could not take place immediately. Here immediately in row 3. Here after 1 minute in row 4. Here immediate. Okay. Now you can see second drop of iodine solutions in row 4 on the sections of tile A, and glass rod was rinsed and dried. Now the step 11 was repeated for 6 more minutes. Now from here step 11 was repeated, this one, for 6 more minutes. You can see here, okay. These are 6 more minutes. This is a second minute. This is third minute. And this is fourth minute. This is fifth minute. This is a 1. This is okay. Second, third, fourth, fifth, sixth minute. And lastly, 7th minute. This is. Now, so step 11 was repeated for 6 more minutes there, okay. And step 13, you can see step 8 to 12. Step 8 to 12, right? These all were repeated for test tube B and B1. And step 14, step 8 to 12 were repeated for test tubes C and C1 also. Now let us see. This is the result. For row 1, we can see this key we can use, right? We can see there is starch. There is starch, okay. So it will turn blue-black with iodine, okay. So there is starch. And second, there was no starch in row second. Row 1 fine, there is starch, but row second there is no starch, only amylase solutions was there, right? So right, the color will remain light brown. Here for row 1 it will be dark blue, okay. For row 3rd, and row 4th, we find there is starch. In both the drops there is starch. Starch is there, right? So as you can see here, for row 3rd, right? So what was taken? Glass rod was then touched onto surface of first and second drop of iodine solutions in row 3. Okay, and the mixture was immediately taken, mixture of starch and amylase. So digestion could not take place, right? So starch is still there. So in row 4th also there is starch, because just after 1 minute, the glass rod was used to remove some of the mixture from test tube A, that has mixture of both starch and amylase solutions. So then touched onto first drop, then second drop of the iodine solution in row 4. So what is the result? No digestions, right? The starch is still there. So this is what? Dark blue. Okay, then we can see row 5. onward, row 5 onward. Let me take different color. Yes, row 5 onward, right? So these all the rows are there, right? How many? 6 rows are there further. These 6 rows. After this, what you find? There is no starch. There is no starch. It means starch has been digested completely, right? So let's see. Okay. Similarly, we can see for test tube A had 3% amylase solutions. A had 3% amylase solutions, right? So trial 1 and trial 2, we can see in both cases, time taken for all the starch to be digested is 2 minutes. 2 minutes. Within 2 minutes, the starch is completely digested. This is after 1 minute, okay? And then this is after 2 minutes, 3 minutes, 4 minutes, 5 minutes, okay? So then you can see. So after 2 minutes itself, after 2 minutes, after 2 minutes, 2 minutes, we can see starch is completely digested. For test tube A. Okay. Now we can see the result for time taken for second test tube B. Test tube B, you can see here there is starch in first row. There is starch. Okay. So dark blue, black color will be seen when starch will contact with the, okay, and row 2 there is no starch because uh, there was only amylase enzyme solutions. Okay, when comes in contact with the iodine solution, no change, so no starch. Okay, and then we can see here row 3, row 4 and row 5, these rows, right? So there is starch, there is starch, okay, and row 6, 7, 8, row 6, 7, 8, the drops join together, right? And you can see there is no starch in row 9 and 10. Row 9 and 10, there is no starch. It means starch is completely digested. So we can see for concentration of amylase solutions 2% for this, for trial 1 and trial 2. This trial 1 and trial 2. In trial 1, it takes 6 minutes. This is 6 minutes. Here, it takes 6 minutes to digest starch, while this is trial 1, this is trial 2. Trial 1, trial 2, we can write here. Okay. For trial 2, you can see it takes 5 minutes to digest starch, right? And then for concentration of amylase solutions, 1% concentrations in test tube C, we can see in row 1 there is starch, row 2 no starch, okay, and row 3, row 4, row 5, right, there is starch. So here we find, right, time taken to digest. Here there is a starch is digested at what time? 7 minutes. It took 7 minutes. You can see last row, and result is this. Here starch is digested. Okay. Okay, at 7 minutes for trial 1, but for trial 2 it digested, you can see here, at 6 minutes. So at 6 minutes you can see this 7 minutes, this is the 6 minute. 6 minute the result is there, it digested, but it is digested before also, you can see, right? So the 7, 6, 5, 4, 3, at 3 minutes also it is digested for trial second. Okay, so results you can write, can draw this table. Okay, concentration of amylase solutions, percentage. So all three percentage you can write here as it is given for A1, B1 and C1. This 3%, 2% and 1% is given. Okay, and there are two trials, trial 1, trial 2. Okay, in per minute, this header line you have to write down. You can take from here the data, time taken for all the starch to be digested per minute. Okay, so results can be noted down here. Now explain why the student results are not reliable. Students results are not reliable. Reason: in B you can see drops at 3, 4 and 5 minutes joined together. For B, the drops at 5, 4, 5 and 6 joined together. You can see 3, 4, 5. Basically, these drops you can see they are joined together, right? This is 3, this is 4, this is 5. At 5, say 3 minutes, 4 minutes and 5 minutes, the drops join together. This could be the reason, you can write here. Okay, now the starch was digested into simple sugars. Simple sugar is what? That is glucose. Okay, describe how you could test the liquid in the test tubes to show they contain reducing sugars. So students, we can add the equal volume of banded solutions, then we can heat the mixture, right? There will be color change from blue, color change, uh, color change will be there from blue. Blue is the color of banded solutions to, right, depending on the concentration of the reducing sugars, right? So it will change from green, yellow, orange to brick red. Now B1, state one variable that was kept constant in this investigations. So you can see the temperature. Temperature throughout it was kept constant. It was 60 degree, you can see the water bath was kept at 60 degree. It is constant throughout. Or you can say there is same volume of starch solution also, that is 5 cc. That also can be written. Okay. So one variable was kept constant in the investigation, that is temperature, you can write. Okay. So how it was kept constant? Use water bath at 60 degree Celsius. That could be done. Now, explain why all the test tubes were left in the water bath. So we can see there are a number of variables. So we can use the temperature. So we can keep it constant at 60 degrees Celsius. Okay. Second, we can write the volume of starch solutions. The same volume was used in each case. Okay. And concentration of starch solutions. The same concentration of starch solutions was kept in each case. Okay. Concentration of iodine or amount of iodine. Same iodine solution used in each. Time also can be kept constant here, right? The 3 minutes for equilibrations, right, or testing for at 7, 8 or 10 minutes. So you can write any one. Okay. Next, explain why all the test tubes were left in the water bath for 3 minutes before the amylase was added to the starch. Reason, so that the contents of the test tubes, all the test tubes, can reach the same temperature. Okay. Explain why step 9 was carried out before mixing the amylase and starch together. You can see step 9, students. This is step 9. Okay. Step 8 was repeated using the amylase solutions in test tube A1 and the drops of iodine solution in row 2 on the sections of the tile labeled A. It was done to make sure that there is no starch in the enzyme solutions. So the color of iodine solution will not change. Okay. C1, identify two sources of error in step 10, 11 and 12. Step 10, 11 and 12, you can see step 10, 11 and 12. Here, a timer was started and the amylase solutions in the test tube. So here you can see the errors you can write here, right? Judging color of the eye is really a difficult task, right? Uh, so improvement can be done that we can use a standard color for comparison, or we can use colorimeter. Okay, so two sources of error I have noted down here. You can see judging color by eye. This is one of the error, right? And second, two samples at the same time are taken. This is also another error, right? For one of the errors you identified in C1, describe how the method could be improved to reduce the error. So we can use, see, like two samples at the same time are taken and we are using the glass rods, right? So two different glass rods should be used. Okay, this can be rectified like this, or for judging color we can use colorimeter. We can use colorimeter, or we can have a standard color for comparison. Okay. D, in another experiment, some students made starch agar. Okay, some students made starch agar that contain 100 mg per cubic centimeter of starch. In 1 cubic centimeter of starch, there is 100 mg mass of starch. So the starch agar was stained using iodine and was cut into blocks that measured 2 centimeter by 3 centimeter by 0.5 centimeter. So we can find out the volume. Okay, so calculate the total mass of starch in each of the block of starch agar. So to calculate it, mass of starch in 1 cubic centimeter cube, right? This is given already. This is 100 mg, right? So volume of one block, one small block, that cut block is what? Cut block of agar, that is 2 centimeter by 3 centimeter by 0.5 centimeter. So it comes to 3 cubic centimeter. So total mass of starch you can multiply. Okay, since the mass of starch is 1 cubic centimeter cube is 100 mg, then in 3 it will be 300 mg. So this you can write here. Now, 6 small beakers containing 20 cc. This is the volume of amylase solution. 5% amylase solutions were placed in water bath at different temperatures. One of the block containing starch from D1 was placed into each of the beakers. Now, the time taken for all starch to disappear was measured. You can see at temperature 20, time taken for a starch to disappear, it is 1500 max. Okay, and gradually you can see as the temperature increases, right? So here you can see the time taken will keep, first it will keep decreasing and then increases. Okay, first decreases and then increases until temperature 60, it decreases and then it increases. Okay, a rate of reactions, you can see rate of reactions will keep on increasing. Initially it keep on increasing, you can see, right? And then, so here you have to calculate the rate of reactions. Complete table 1.1 by writing in the rate of reactions at 70 degrees Celsius. What will be the rate of reactions here? Rate of reactions. So how will you calculate a rate of reactions? Rate of reactions we can calculate by dividing the mass of the starch by time taken. Okay, mass of starch, so rate of reactions at 70 degrees Celsius will be equal to mass of starch is what? 300 mg, and divided by time taken to completely disappear starch is 88 minutes. 88 minutes are taken, right? This much, yeah, sorry, it is in seconds. So time you can write in seconds, it is given already. So when you will calculate, you will get 3.4 mg per seconds. So it is 3.4 milligram per seconds. Now plot a graph on the grid to show the effect of temperature on the rate of reactions. So students, rate of reactions you can put on the y-axis, right, and temperature on the x-axis, because temperature we are changing. Temperature is what? This temperature is independent variable. This is independent variable, right? Whereas this rate of reactions, it is a result, right? This is dependent variable. So dependent variable we always place on the y-axis. You can see y-axis, and the independent variable on x-axis. So you can take a suitable. So a temperature is minimum is 20 and maximum is 70. So accordingly you can take the scale, right? So 0 to 10, 10, 20, 30, 40, 50, 60, 70, 80. So one small grid is equal to 1 degree. Okay, and rate of reactions, yeah, minimum is zero point 0.2 and maximum is 4.6. So you can see here, so 4.6 means here you can take a scale of means one full big one. Okay, big block is equal to 1. Okay, and 1, 2, 3, 4, 5 like that. So 0, between 0 and 1 there are 10 small grids. Okay, so you can start like this, okay, and you will get graph in this way. Initially the rate of a chemical reactions increases as the temperature increases. It reaches up till 50 degrees Celsius, it is increasing, okay, and then from 50 to 60 it increases rapidly, reaches at peak. Rate of reaction at peak you can see, okay, it is 4.6, and then after this it will start decreasing. Now question number 2, figure 2.1 is a photomicrograph of epidermis of a leaf. It shows epidermal cells, guard cells and stomata. You can see each stomata is surrounded by two guard cells containing chloroplast. So you can see here, this is the stoma, cytoplasm, okay, and cell wall. You can see guard cells. These two guard cells are there, kidney shaped. These are epidermal cells which are irregularly, okay, arranged. They are larger one also, and then you can see chloroplast lying in the guard cells. Now complete A1 complete table 2.1 to show two visible difference between epidermal and guard cells. So my dear students, you can see this is the epidermal. This is the epidermal cells and this is guard cells, right? Guard cells. You can see the difference between these two, right? Feature like you can see chloroplast. Chloroplast and cell wall, these two features you can take, right? Chloroplast is absent in epidermal cells, okay, while in guard cells they are present. You can see chloroplast are there, many chloroplasts are there, but in epidermal cells, these epidermal cells, there is no chloroplast here, okay. And cell wall if you see in epidermal cells, it is thin cell wall, while in the guard cells you can see the cell wall is thick, and in epidermal cell, right, the guard cell is cell wall is thin. Okay, now make a large drawing of the two guard cells and the stoma shown inside the box on figure 21. You can see, oh, this is the box is given here. These two guard cells you have to draw. Something students you have to remember, this I have drawn, for example, sample only, right? Your drawing should be clear and continuous. The line you are using, that should be clear and continuous, right? And the image you are drawing, the drawing should be large enough. It should cover okay at least 60% of the space provided. Now, and this should be a double line for cell wall. For cell wall you can see this is double line, right? And there is stoma, is the stoma you can show, okay, and there is no shading is required. These all points you have to follow, students. This is more important, okay. These all 5 points you have to follow. Labeling is not required to label the your drawing now. Part B, measure the total width of the guard cells and stoma along the line XY. Along the line XY, it is already given there, students. You can see, right, in the image. Here it is given. This line you have to measure with the scale, right? And after this, you can write it will come around 31 in this range, 31 to 34 mm, and draw a line on your drawing in the same positions as the XY, right? So that you have to measure. You will be drawing in your paper, okay? And then that measurement you will be writing here. The values you can apply and to calculate the magnifications. Width of on your drawing and width of the figure 2.1. Figure 2.1 it is given only. Okay, you can measure with a scale. Okay, and then you can calculate and you will get your answer. Right. C part. The figure 2.1. Figure 2.1 shows the rate of water gain by absorptions and rate of water loss by transpirations. Water gain by absorption and water loss by transpirations in the plant during 24-hour period on a hot sunny day. You can see here. So here for absorption it is shown by dotted lines, right? So initially you can see from 6 to the time you can compare this. Absorption, rate of absorption is lower than rate of transpirations, okay, between 9 to 18. 9 to 18 you can see here easily. I will just simplify it. Okay, you can see between 9, so this is 6, this is 10, and then 4. Okay, there are 10 blocks, so you can see here 9 from here, you can say this is 9 from here, right? So rate of absorption is lower than the rate of transpirations, and it is up to until 18 time of the day, right? From 9 until 18, the rate of absorption is lower than rate of transpiration. Rate of transpiration is higher. Rate of transpiration is higher here. Okay, then we can see second point we can write here. Rate of absorption is higher than rate of transpiration from 18 till 16. 6, sorry. From 18 till 6 you can see rate of absorption from here up till this, right? So rate of absorption is higher than rate of transpiration. So whatever you see in the graph that you have to write. Okay, D part, figure 2.3 shows the apparatus used to measure water uptake by a leafy shoot. The leafy shoot is sealed tightly into a glass tube which is connected to a capillary tube containing water. So a leafy shoot loses water through its leaves. It absorbs water from the apparatus. Air is pulled into the open end of the capillary tube as the water moves towards the leafy shoot. The distance moved by the air in the capillary tube can be measured, okay, on the scale and used to calculate the volume of water absorbed by the leafy shoot. So here you can see this apparatus is called photometer. Okay, this is used to measure the rate of transpirations, that is loss of water as vapor by the shoot. So what will happen as the water will be lost? Okay, so the bubble, the air bubble will move ahead, and we can measure the distance covered by this air bubble, right? Because the this root part, okay, the shoot, cut end of the shoot, right, will absorb water, and this water will be lost from the leaf part. It will be lost, right? So water will be moving here. So due to this, the air will also move ahead. So distance covered we can easily calculate. And then air bubble we can send back by turning this tap on, this knob on, right? So that from this reservoir water can push it back. So describe how you would use apparatus, right, to investigate effect of temperature or humidity, to either temperature or humidity on the rate of water absorptions by a shoot. So students, these are the points you need to remember, right? Like point 1, you can see, take 3 apparatus as shown in the figure 2.3, means 3 potometer you need, right? Okay, so that we can keep them at 3 different conditions. Okay, so put each apparatus at 3 different temperatures, say 20 degree, 40 degree and 50 degree Celsius. This is what just examples. Okay, and then here control variables are same light intensity was given, same wind speed or humidity. Now use the same plant with same number of leaves. If we are using different plant species or number of leaves variable, then it will affect the result. Okay, measure the distance traveled by air bubble in the capillary tube, because that is already graduated. Okay, and this is mandatory. Cut the stem underwater. If you cut in the air, then chances are there the air can enter, that can interfere with the absorptions. Okay, so cut the stem underwater and dry the leaves. And this is more important, repeat the experiment and take the average. So here independent variables are what? The conditions, temperature, okay, 3 different temperature, that is the independent variable. Dependent variable, that is the result, rate of absorption of water or distance traveled by the air in the capillary tube. And repeat, it's important. And keep the what is the improvement? Improvement, you can see, the cut the stem underwater. So these 6 points you can write to get any 6 point. Okay, you can write to get full marks here. Thank you, students.